Good evening, Mr. Matthew, and everybody who is watching us. Today, we're going to present the last laboratory practice, the laboratory number 6, which we have to do a report. Our aim for this topic is air microbiological sampling as a management tool, which is presented by David Quito and Wilmer Illescas. So let's begin with.
Yeah, in this case, we are going to talk about the contents, the presentation, and there are introduction goals, methodology, discussion, final question, results, and discussion of the results, and conclusion. How introduction? Editor 2024 mentioned that microbiological analysis is crucial to evaluate the air quality in all facilities and understand the microbiological risk associated with possible contamination. This information allows us to develop effective strategies to control the presence of microorganisms in the environment. Additionally, this type of sampling is essential to verify the effectiveness of our cleaning and disinfection procedures. The atmosphere does not have an indigenous microbiota, but it is a medium for the dispersion of many types of microorganisms, fungal spores, bacteria, and viruses coming from other environments. Our goals are general, understanding the significance of microbiological air sampling in environmental quality management within facilities is pivotal for developing practical skills in sampling techniques and result analysis, ensuring a safe and healthy environment of occupants, implementing and quality standards and safeguarding public health. And the goal specific is understanding the importance of microbiological air sampling in environmental quality management within facilities, and cultivating practical skills in techniques for microbiological air sampling and results analysis. In this case, the practice only used materials and instruments that we needed. Petri dishes with selective culture medium, Scotch tape, and a specific place where the Petri dish will be left. Our methodology is of this practice, letter A, identify the area to be sampled, in this case was Krispan, and ensure it is clean and clear. Place the Petri dishes in strategic locations to capture the greatest number of microorganisms in the air. Letter B, open the Petri dishes and place them in the open position in places where they can receive ambient air directly. Make sure the plates are level and stable. Letter C, expose the plates to air for the desired exposure time, which can vary from 1 hour to 1.5 hours, depending on the level of contamination of the area and environmental conditions. Letter D, after the exposure time, close the Petri dishes and seal them with adhesive tape. Label each Petri dish with the location and exposure time. And finally, of the methodology, place the Petri dishes in an incubator at the appropriate temperature and condition for the growth of microorganisms for the time necessary for colonies to develop. Generally, this growth is 24 to 48 hours, and count the UFC.
All right, so now we're going to present the results that we have gotten. In this case, our results, as we can see them, we have the microbial count. Well, basically, my partner said that we had to put 2 Petri dishes in a place, in this case, Krispan, which is a place where it's like sort of a shop that is here at the university. So we put both of them, we had 2 samples, sample 1 and sample 2, and we put them in different places. The sample 1 was put in a place which is much more crowded by people. And the sample 2 was in a sort of a shelf book in a place like where many products are, but sometimes nobody passes. So no, it's not usual that people passes throughout that place. So after we went to the laboratory and counted these results, we found that we had bacterial colonies 84 in the sample number 1, and the fungi we had just two fungi species. So you can see here, for example, that we have this one, which is the first fungi, and the other one, the second one. And all of these little spots, blue spots, are the bacterial colonies that we counted. On the other hand, we have the sample number 2. Here we can see that we have the same, just one fungi, as you can see here, as well as many bacterial colonies. So you might be wondering what is going to be like the sort of the analysis. Why is it happened? Why did we have more colonies of bacteria in sample 1 rather than in number 2, and the same with fungi? So let's analyze that. Here we have the analysis of the results. Firstly, what we did was, well, sort of the same. We just took the data we had here, and then we put them in this place. UFC is sort of like CFU, like the unit formation colony. So with this unit formation colony, we just put it 84 and 83, the same. Why did we put the same? Because actually, remember that this is like the cell that made that propagated in order to create a colony. The time that we did this experiment was 2 hours. Therefore, we put it here 2 hours. And then here we have the UFC over hours. And here you can see that we have like a rate of 42 UFC per hour. It means that we got 42 bacterial cells per each hour. And well, here is the same, like just in minutes. UFC in per minute, 0.7. Basically the same with the sample number 2. We have 84 and 2, the same thing. The only difference is like the rate of change was like is slightly slower, and the same here. Yeah, there's like not too much difference. Now let's analyze the fungi analysis. Let's analyze. This is interesting because in sample 1 we got 2 and sample 2 1, but the time was the same. Actually, it was like per hour we got one fungi, and on the other hand, in sample 2 we had just 0.5, like the median of a fungi per hour. It means that in the 2 hours we just got one fungi, and here's just the time. It's like a so tiny number. As you can see here, we can see that the sample 1 shows a slightly higher concentration of microorganisms. That was due to the fact that we put it in a place where more people usually pass. More people maybe could have a cold and then just say they sneeze in that moment, and then all of those microorganisms just are propagated into our medium. Finally, let's present the conclusions, which are the ones that we're going to end with. So, to end this presentation, let's review what we said. Firstly, well, my partner told us that we went to Krispan, and then we put 2 samples there. Our aim was to analyze how many bacteria or overall microorganisms we have in a medium. So with that things that we have, with those microorganisms, we did the analysis, the calculus, as you can see here. All right, so that was what we did. And as you can read here, it says both samples suggest that there is a normal concentration of them in this area. Yeah, because imagine that we could have gotten like a lot of microorganisms there, like more than 245, which is the maximum amount in order to count the microorganisms. That would have been odd, because whether or not we had this amount, it would have been like much more difficult to count. We would have to like dilute the concentration, and then we could find the proper amount of cells. Well, so the number of microorganisms were all right, were like stable, the number amount. Then we saw that example one had slightly more organisms than sample two. That was due to the fact, as I mentioned before, like we put sample one in a place which was much more crowded. Yeah, and finally, it says that, well, both samples show nearly the same rate of change in the number of colonies per minute. All right, so that's true, owing to the fact that, well, I showed you before, like we have like the same rate here, UFC per minute. So, well, basically, this is our exposition. Thank you so much for listening to us. We will see you in a next opportunity, maybe in the next semester because of that presentation. And we say, David, we say, this is the bibliography. Yeah, just in case. We have the bibliography there. And we say, David, we say,
You what?
thank you. We say thank you.
Thank you very much.
Bye.